Welcome to ChemTutorial 1.1, and in this tutorial we're going to be talking about the scientific method briefly, and then we're going to spend most of the time in this tutorial talking about lab report writing. How do you write a lab report, and how do you develop an experiment? So first I'd like to talk about the father of Western medicine, and his name is Hippocrates. Hippocrates lived between 460 and 370 BC, so he's a very old guy, and he's the person who really kind of started this process of observing the natural world and making conclusions. The problem with Hippocrates was all he did was he saw something, okay, that was happening in the world, and then he thought about it, and he made a conclusion based on just the things that he saw. So the problem with his way of thinking was he just used logic and reasoning to determine truths without having any kind of real experiment to base his conclusions off of. So eventually, several hundred years later, we've moved on to having a more in-depth way of observing the natural world. After we observe something in the natural world, we create a hypothesis, and that's just basically it's a tentative explanation of how something works. Okay, Hippocrates and early scientists would stop at the hypothesis section, and they would call their hypothesis fact. But what we now know is that we take our hypothesis and we're actually going to do something about it. We test it through an experiment. After we conclude our experiment, we then observe what happened. As a result of that, we record the results, we make a conclusion, and then we start the process over again. So the process of the scientific method and experimentation is a cyclical pattern that we continually cycle through. There are three main types of experiments, and primarily in our class we will be doing the experimental experiments, but I want to talk about all three types. The first type is a descriptive experiment. That is simply something where we observe something that happens and we describe it. So I observe that water is clear and I make my conclusion. So these types of experiments are just based on what questions. So for example, if I asked you what is the color of water, you would observe and you would say it's clear. So these are very low level thinking types of experiments. They don't really require a lot of critical thinking at all. They're just really observations. Comparative experiments are definitely more higher level. It's when you take two different groups. You have an experimental group and another experimental group, and you essentially do something to those groups and then you compare the results. Okay, so these types of experiments are usually based on which questions, such as which candy gives you more energy, or which soda has more calories. So they do use critical thinking, but they're not the highest level of experimenting. The highest level of experimenting are the experimental experiments. This is when we actually create a hypothesis, you test it, you provide concrete evidence to either support or not support your hypothesis, and then you generate an explanation for a phenomena. These types of experiments are created to answer a how question. There are several steps that you have to follow in order to create an experiment. Okay? The very first step is to ask yourself this question: is my experiment testable? Okay? That's step one. Step one. You should write that in your notes. Step one is to ask yourself that question: is this experiment even testable? The way we know whether an experiment is testable or not is if there is a way for us to get quantitative data results from the experiment. What does that mean? What does the word quantitative mean? Well, the way I remember what quantitative means is I know there's an N in quantitative, and the N stands for numbers, meaning you have to have some data that corresponds to numbers. For example, an example of quantitative data would be I have 6 boys in my class and 12 girls. Okay, so I'm using numbers to represent some concrete piece of information. Qualitative data is descriptive data, where you're describing what something looks like. Okay, and so the way I remember the difference between qualitative and quantitative is that qualitative has an L, and so that makes me think of the word look. And so qualitative data is describing something, specifically what it looks like, okay? So here's some example experiments, and let's figure out whether they're testable experiments, whether they're not testable, or whether they could be testable, but we need more clarification. Okay, the first one says, my dogs are louder than your dogs. Is that a testable experiment? It depends. If I'm taking my opinion on whether a dog is louder than another dog, then no, it's not testable, okay? Because opinions are not testable. That's an important point, something you want to write down. Opinions are not testable, okay? However, if I had a instrument, if I had some sort of technology tool that was actually able to measure the loudness of sound, then could that be testable? Yes. So I would say this is testable, but it needs clarification. What about the next one? My dogs are more annoying than your dogs. Well, annoying isn't something that we can actually test. We can't get a number on how annoying something is, okay? If I decided to make an annoying scale from 0 to 10, that would be my opinion, right? So since this is an opinion, it's not testable, okay? Now the next one, my dogs make more neighbors move than your dogs. Is that testable? Yes, I can actually count how many neighbors are moving, right? If my dogs made 2 neighbors move and your dogs made 1 neighbor move, then that's a testable experiment. So yes, it is testable. What about if I said my dogs are more frightening? Again, frightening is like annoying. It's an opinion, so this is not testable. My dogs scare more people than your dogs. This one can be testable as long as we're not rating the level of scariness. If we're actually counting the number of people that are scared, it's testable. Okay. Next, so that was step 1, is your experiment testable? The next question, so this is step 2. Okay. Step 2, you need to ask yourself 2 questions. What are you testing? And how are you measuring the outcome? Okay, so what are you testing and how are you measuring the outcome? So to answer these 2 questions, you need to know 2 definitions. And I'm sure you've heard these before, but I want to review these definitions anyway because we use them a lot in chemistry. The first is independent variable. An independent variable is the thing that is being manipulated. The way I can identify what my independent variable is in my experiment is if I ask myself, what is changing in the experiment? Okay, what am I actually changing? What am I doing? Let's say I was testing how tomato plants respond to different colors of light. Okay, so I have multiple tomato plants and they're all the same tomato plant. Okay, they're all the same type of tomato plant. And one of them I exposed to regular sunlight. And the other one I exposed to red light. And the one after that I exposed to green light. Okay. And so they all have different colors of light. And I'm measuring to see which one grows, what light helps the tomato plant to grow better. What am I actually changing in the experiment? I am changing the light, right? So that is going to be, what are you testing? I am testing light. I'm testing which light works best on the tomato. The second part of our step 2 question was, how are you actually going to measure your product? How are you going to measure your result, the outcome? And that is where the dependent variable comes in handy, because the dependent variable is just the thing that's being measured, okay? So this is the question you want to ask yourself if you have to identify what a dependent variable is. The question is, what are you actually measuring? So in my example with the different colors of light in the tomato plants, what would I be measuring? I would be measuring the height of the tomato plant, right? Because I want to measure which tomato plant grows best in which light. And so I can measure growth by using a ruler and measuring, how many centimeters each tomato plant grew in their respective light. So in the example I gave, it would be the height of the plant. Okay. So now that you know what you're going to be changing, what you're testing, and you know how you're going to measure that, okay, so you know your independent variable and you know your dependent variable, then you can move on to step 3, okay? And step 3 is where you plan out the conditions for each of your groups that you're testing. And you limit variables. Step 3. So let me talk about why those things are important. There's 2 types of groups that you can have when you're doing an experiment. You can have a control group and you can have an experimental group. Not every experiment is going to have a control group and an experimental group, okay? Some experiments will only have experimental groups. An experimental group is an object that you're changing something on, okay? So in the experiment that I was mentioning, each experimental group would be a separate tomato plant in a different color light, okay? So the tomato plant in green light would be one experimental group. The tomato plant in red light would be another experimental group. The control group is just going to be your normal group, or the one that you don't change anything on, okay? So my control group in the experiment I was using as an example would be the tomato plant in the regular sunlight, right? Because that's the normal way a tomato plant grows. In order to test a hypothesis, what you also have to do is you want to remove any ambiguity, okay? Any ambiguity from the results by designing an experiment that is controlled. Okay, so what do I mean by that? Well, in the experiment with the plants and the light, I only want to have one variable that I'm changing in the experiment in order for it to be an accurate experiment. So for example, I'm not going to want to change the soils in the tomato plants, right? And I also don't want to change the type of plant, because if I change multiple things, then I'm not going to know what the result is due to, right? If I leave everything the same, if I leave the temperature of the plant the same, if I leave the soil of the plant the same, if I leave the water the same in all of the plants, and the only thing I'm changing is light, then when I look at how the plants have grown, I know it's only due to the light, right? It's not due to the fact that I did anything else differently. That's really, really important. So that's what we call constants. That's things that are the same in every single group. And you'll want to write that down. Constants are things that stay the same in every group. Okay? You only want to have one variable, and that's your independent variable. That's the thing that's you are actually changing. Okay, so now that we're ready to start our experiment, I want to walk you through how you write a hypothesis for this experiment and how you write an objective. So the criteria for a hypothesis is that obviously it has to be testable, like we talked about earlier, but we write our hypotheses as if-then statements, meaning that I start off my hypothesis with the word if, and then I end my hypothesis with then, okay? So in my hypothesis, I have to say what my independent and my dependent variables are, and then I have to describe them. And then lastly, I have to say how the independent and how the dependent variable are changing. Okay, so this is your sample form for writing a hypothesis. Let's write a sample hypothesis for the experiment with the tomato plants. I would see if the, and then what's my independent variable? What am I changing in the experiment? I was changing the light, right? So if the light, and then how did the light change? Well, I changed it to green and I also changed it to red. So I'm going to say if the light changes color. And then I write then the, and what was my dependent variable? What was I measuring? I was measuring the height of the plant. So I would say then the height of the plant will, and then I describe what I think is going to happen. What do I think is going to happen to the height of the plant if I change the color? Well, let's just say that I think that green light and red light will make plants not grow as well as normal sunlight. So I would say if the light changes color, then the height of the plant will decrease. The last thing I want to show you is how to write an objective for your lab. An objective is essentially the writing down the specifics of your lab in 1 or 2 sentences, okay? So these are the things you have to have in an objective. And when we do labs, I will have you write objectives for some of the labs to practice this. The first thing you should always include inside your lab is how you're going to change the IV. So for example, you would have to say something like, I will change the light color to green and red. then you would want to say what your expected result is. For example, I would say I'm expecting the potato plant, the tomato plant to not grow as well in red and green colored light. Then you describe the variables in your control group. Remember, the control group is the group we don't change. So for example, the tomato plant in regular sunlight, okay? So my variables would be, I have a tomato plant that's in the regular sunlight, okay? And then I have 2 other tomato plants in red light and green light. And then also list all the constants in the experiment. And you don't have to list all of them because really there's like a million constants that I could come up with. But some of the constants in my experiment might be the fact that my soil is all the same, the fact that my watering is all the same, maybe the humidity is all the same, okay? Things like that. And that is it for our ChemTutorial 1.1. You may now go ahead and work on the skill check and have a great day.